All right. Welcome, everyone. Today, we're going to be talking about networks. So this is the first of 2 lectures on networks. Here we are in the middle of module 3. We talked about regulatory motifs, which is one way to define regulatory networks based on connections between regulators and their targets, and even their targets and their target regulatory regions and the genes that these regulatory regions target. Today, we're going to be talking about how we can actually study the emerging networks, whether they are protein-protein interaction networks or regulatory networks or functional relationship networks. And then on Thursday, we're going to be talking about deep learning. So for today, we're going to first introduce what networks are, what kind of networks emerge in biology, and how do we study them, or what are the different sort of computational formulations for these networks. Then we're going to talk about measures of network centrality. You very often want to use the network to find nodes that are important based on their connectivity relative to their neighbors. We're going to talk about linear algebra ways of studying networks and decomposing networks and breaking networks up. Is the sound coming through? Perfect. And then we're going to extend that into principal component analysis and specifically sparse PCA, which allows us to now start finding the more important components of a network through its global properties. Then we're going to talk about network communities and modules, how we can actually decompose networks into similarly functioning components, and how we can use that to infer the function of nodes based on their neighbors, also known as guilt by association. And then we're going to talk about how information spreads through network, so network diffusion, and the opposite of that, which is figuring out which edges are direct and which edges are actually indirect from that diffusion. So let's jump right in. So living systems are organized in this hierarchical fashion across many layers. So at the lowest layer, you have genomic information. Then you have epigenomic information sitting on top of it, transcriptomic information, which is modulated by the epigenome, and then proteomic information. And at every one of these levels, there are a lot of interactions between these components. So you can think of these interactions as happening within each of these layers, whereby different chromatin marks might be interacting with each other, different genes might be interacting with each other, different transcripts, different microRNAs, different proteins. And you can think of these interactions at each of those levels. So at the level of metabolites, for example, you can have networks that basically describe which metabolites are translated and transferred and sort of into what other metabolites. And of course, the ones catalyzing these metabolic reactions are in fact enzymes or proteins, which basically interact with each other at this protein level and signaling level. And then what transcribes, what makes these proteins, basically regulatory networks that control how these proteins are made and when and where and what cell type. And then what controls these sort of post-transcriptional and pre-transcriptional regulatory elements. These are the motifs that we learned about and the transcription factors that we learned about and the microRNAs that we learned about in previous lectures. You can basically think of this dynamic transfer of information from genome to RNA to proteins to ultimately the dynamics of these emerging properties, and then interactions happening within each of these layers, but also interactions happening across layers. So there's multiple different types of biological networks. We've talked about regulatory networks in the previous lecture. This is when transcription factors interact with particular regulatory regions, and those regulatory regions control the activity of particular genes. You can have inhibitory interactions or activating interactions. You can have, signed interactions. So basically every single one of them is between a regulator and its target. And the target does not connect to the regulator unless, you have feedback loops. So these networks are directed, they are signed, and they're also weighted. So they can be positive or negative. They're always directional from A to B, but not necessarily from B to A. And then they're weighted where the strength of each interaction represents the magnitude of regulation. Everybody comfortable with the first column here? Yes? So metabolic networks are basically between metabolites. So, you know, this particular metabolite can be, you know, or basically changed into that other metabolite and so on and so forth. And then the agents that are responsible for these changes are proteins. They're enzymes, and they're sitting at the edges. So here, the proteins and the genes were sitting at the nodes. Here, the proteins and the enzymes are sitting at the edges. And these networks are very often bidirectional, where every enzyme can be, every metabolite can be, you know, transferred back and forth, if you wish. And they're usually weighted based on the ability of every protein to carry out that interaction. Signaling networks are basically about a layer of gene regulation that happens without the involvement of DNA. So you can have receptors at the surface of a cell, or receptors at the surface of the nucleus itself, with signals that are being received that then launch a cascade of information transfer all the way to a particular transcription factor, which can then turn on particular genes and so forth. So these networks are directed, and they're usually unweighted. Basically, it doesn't matter how much of that protein is there. If you phosphorylate this protein, it's a binary switch. Protein-protein interaction networks are the basis of protein complexes, whereby these two proteins are known to physically interact with each other. And then these guys are undirected and unweighted. We don't normally have the level of interaction. We just know whether two things interact or not. And then functional networks don't have any biological interpretation as, you know, a binding event or a, you know, a biochemical change or a, you know, signaling cascade or a physical interaction. They are instead networks of co-expression, functional networks that basically say these two genes tend to co-occur in the same cell types or tend to be co-expressed and so on and so forth. But this is another very, very common type of network that we're going to be analyzing. These networks are usually undirected, and they're usually weighted. So because we, you know, if D is correlated with F, we don't know if the direct, I mean, there's just simply no directionality. They're correlated in its symmetric relationship. Yeah. Yeah. Sure. Given additional biological information, we could weigh them. So basically, for the level of information that we have now, they're usually unweighted, but with additional information, we could weigh them. Any other questions? Okay. All right, so there's a lot of information exchange across these networks. As I mentioned earlier, signaling networks ultimately activate transcription factors, which then lead to the transcription of proteins or specific enzymes, and then these enzymes catalyze metabolite signaling changes that can then feed back into a particular signaling network based on the amount of a particular metabolite. The proteins themselves that are transcribed interact with each other, and then they form complexes of transcription factors or chromatin factors, which can then modulate these steps and so forth. So basically, these layers that I mentioned earlier interact a lot with each other. So there's a lot of applications and challenges associated with network analysis. So basically, number one, how do you even identify these elements, the basic building blocks of these networks? What are all the regulators? What are all the regulatory motifs? What are the target genes? So we talked a lot about that in the motif finding lecture. How do we use networks to predict cellular activity? So that's where a lot of systems biology falls in terms of differential equations that you can use to predict flows of particular metabolites and levels of expression of a particular gene. So you can predict the expression level, you can predict the gene ontology or the functional annotation of a particular protein based on these networks. You can infer networks from functional net data. For example, if you have gene expression patterns, as we showed in lecture 5, you can then use those to infer the structure of the network of what proteins are co-expressed or correlated. And if some of them are transcription factors and some of them are only target genes, you can basically build directions there from the activity pattern. And you can also infer the function of these networks. So certainly X is targeted by both A and B, but you'd like to also build a function that predicts the level of X based on the levels of A and B. So this particular function is not just a connectivity, but it could be 3A plus 1B or something, or minus 3B or something like that. So this particular function is yet another level of inference. And then we can also analyze the structure of these networks, basically recognize hubs, like highly connected nodes, or network motifs, patterns of connectivity that are recurrent, or specific functional modules by decomposing these networks. Everybody with me so far? Great. So probabilistic networks are a specific class of these kind of functional networks, and they have very specific interpretations. So, you know, many different types of probabilistic networks exist. Each has different meanings and different applications. And in these networks, these basically represent graphical probabilistic models where the nodes are variables and the edges represent relationships between these variables. So that allows you to now start modeling the joint probability distribution of many variables using graphs that tell you how information flows within these networks. So for example, in Bayesian networks, these can be directed. In Markov random fields, these are undirected. And in general, when you have these types of edges, the most important part is the edges that are not there. Because these edges basically tell you that there's simply no information flow between this particular node and that particular node directly. If I condition on this particular node and that particular node and that particular node, for example, then there's absolutely no information that can flow from this node to, say, that node. So what edges in these probabilistic networks usually mean is conditional independence. So for example, if I draw edges this particular way, and all of the information between S1, group S1 and group S3 flows through group S2, that means that once I have observed the values of S2, it doesn't matter what I tell you about the values of S1. This will have absolutely no impact on your estimate of the values of S3. Raise your hands if this was clear. Awesome, great. So if I don't specify S2, then of course, if I learn that this value is huge, then that information will flow down, tell me something about the levels of expected values and the distribution of these values for S3. And I can vary any of those, and through these connections, information will flow. At the moment I've observed those, I've broken the flow of information, because anything I'll ever know about S2 has been known already by observing S2. So it doesn't matter how I change the values of S1. So this is the difference between physical and relevance networks. So physical networks, the edges actually represent some kind of physical interaction, such as a regulatory network that has a physical binding between a transcription factor and a regulatory region, a physical looping between that regulatory region and the gene, and then a physical transcription between the gene DNA and the mRNA of that gene, and ultimately translation of that gene. So all of that has some kind of physical instantiation. Whereas relevance networks, these functional networks, if you wish, the edge weights simply represent similarities between the nodes, or a functional relationship between the nodes. And this could be something about co-expression patterns or co-regulation patterns. So how do we represent networks mathematically? Well, we represent them as graphs. So these can be weighted graphs, where there's a weight associated with every edge, and generally these weights can be positive. Sometimes they're negative. They can be multi-graphs. These are not true graphs, they're pseudographs, where multiple edges exist among the nodes. So A to A, for example, might exist there twice or 3 times, and so on and so forth. And this could represent just different paths of going from one node to another node. For example, in a road network, you may have 2 different roads starting from the same point and ending to the same point. You can have directed graphs, also known as digraphs, where the edges are actually directional. And you can also have simple graphs, where there's no multiple edges or self-loops. Basically, any kind of self-loop is eliminated, any kind of multiple edges are flattened out, and then these are simple. Okay? There's some basic definitions of graphs. How do we represent them computationally? So if you have this graph representation of a network, there are two dominant representations that you can have. One is a matrix representation that basically tells you from every node to every other node whether there is an edge or not, and what is the directionality of that edge based on whether you are B to A or A to B, will be here, on whether you are on the upper or lower diagonal, triangle here. And then the signing, whether it's positive or negative, will be represented here, and then the weight will be represented there. Okay? So this is the simplest representation, the one we're going to be using the most often. For extremely sparse networks, and now an alternative representation is the adjacency list representation, which basically tells you the list of partners for every node. So A, for example, will have an adjacency list of B, 1.5. B will have an adjacency list of A, 1.5, C, 3.4, and so on and so forth. And that can be a very compact representation for extremely sparse networks, but it's not as easy to compute with. Instead, the matrix representation is very easy to compute with, and it also allows for linear algebra operations, for example, matrix multiplication, which would represent taking, basically going from A to B and B to every other node. You can just represent that very simply by squaring that matrix, for example. And then in this representation, the degree can be a weighted degree. For example, if I want to sum up all of the weights incident on node C, I can just simply add all of those up, and then this is what I get. I get degree 6. Okay? Everybody with me so far? So these are just basic network definitions. So we talked about different network types, regulatory networks, metabolic signaling, interaction networks, functional networks. We also looked at algebraic ways of computing on these networks, and then Bayesian interpretations of the conditional independence that is described by these networks. Let's now switch gears. We're going to talk about network centrality. So what you would like to know is if I have a particular biological network, and we can think of, I don't know, a protein-protein interaction network, or a functional network of co-expression patterns, then I'd like to know how important are different nodes in my network. So how important is a particular node, or how important is a particular edge to the structure of the system? For example, if I knock out this gene, will the network be impacted heavily? Will the function of the cell change a little or a lot? And one way to do that is to think about the connectivity patterns. And what are some examples that you could use? You could use, for example, the node of a particular, the degree of a particular node. So that can basically tell you this particular node has many, many connections. But this particular node only has 3 connections. And yet, if I perturb this particular node, if I knock out this particular gene, I might lose connectivity between this subnetwork and that subnetwork. So this is a low degree node, which is actually very important. And for example, this node here, even though it's connected to many, many different other things, if I knock it out, there's still many paths through which information might flow. So the effect of a very highly connected node might actually be lower than the effect of a poorly connected node. So we'd like to develop methods, computational metrics, for dealing with these different intuitions. Everybody with me? Awesome. So one such metric is simply the degree centrality, or simply the degree. It basically tells you how central is a particular node based on the number of nodes that it is directly connected to. So in this particular example, node B has 4 neighbors. We're going to say, okay, great. Node B appears to be the most important in terms of degree centrality. Okay? So degree centrality can be very easily defined for, these simple graphs, for directed graphs, for in degree and out degree separately. So, depending on the number of edges that are pointing in or that are pointing out of a particular vertex, you can, define separately both the in degree and the out degree. And some nodes will have very strong out degree. These will be master regulators that control many, many other genes. Some of those nodes will have high in degree. We think of those as master integrators that basically sum up signal from many, many places. And then, this can be similarly defined for weighted graphs and, weighted degrees. And stuff. Okay, ready with me so far? So the other measure of importance is that there's a lot of information that flows through that particular node. So this is another measure of centrality that we're going to define as betweenness centrality. So one way to define that would be to say, what is the number of shortest paths that go through my particular node? So in this particular case, all of the shortest paths between any of this group and any of that group, for example, will, go through one of these, 3 nodes here. So that basically suggests that these nodes have very strong betweenness centrality. You can basically count the number of shortest paths from any node to any other node. So this is the number of shortest paths from I to j, or I different than j. And then you can ask, what fraction of those paths go through node k? So basically, the number of shortest paths that go through I, from I to j through k, versus the total number of shortest paths. And that you can think of as the betweenness centrality of node k. Okay? So nodes with a high betweenness centrality control information flow in a network, and the edge betweenness can be defined similarly. Basically, how many shortest paths go through a particular edge. Okay? Everybody with me so far? So in this particular example, the shortest paths are, AB, AC, ABD, ABE, and so on and so forth. And then you can ask how many of those go through node B. 1, 2, 3, 4, 5 out of, 10 possible shortest paths. So that basically gives you a betweenness centrality of 5 for node B. Yeah. Sorry, A, B, B, C. So we're looking for nodes that are different from J, basically. So all of the paths from edges that are not J itself. And from one node and another node that are not the same to each other. Does that answer your question?
Yeah, but why doesn't it count as node this path that goes to the B?
Yeah, I mean, you could, again, there's been so many network papers written that almost every such metric can be altered to include, different boundary conditions, different metrics, different ways of combining metrics, and so on and so forth. What you're suggesting might be a good idea for some scenarios. Yeah, absolutely, you could do that. I mean, what you have to always think, basically what I'm giving you here is a set of tools. You guys have to think for every application, which of these tools, apply the best in my case based on my biological intuition of the problem. Yeah. Yeah, you're right. There's a discrepancy between this particular equation and that particular sum. I agree. You're right. Yeah, so maybe, this should be I different than J, comma, J different than K or something. Anyway, or actually I different than K and ignore J. So that's sort of the definition that's consistent with this particular equation. Yeah. You're right. We're going to have, one way to do it is to weigh basically if there are 3 such paths and one of them goes through, basically give it a 1/3 or something. Yeah. Good question. Other questions? All right, so that's betweenness centrality, basically how many paths go through the particular node. Another way to define centrality is to basically define how close is that node to all these other nodes. Basically, if I take shortest paths from this node to all the other nodes, just how proximal is it? So you could basically count the distance between I and J for every pair IJ, and then ask just how close is, this particular node to all the other nodes. So basically, node B in this particular case is the most central one in proximity as well. So you can basically say that, you know, the distance between B and all the others are 1, 1, 1, 1, as opposed to C, which is 2 away from here and 2 away from there, and A, which is 2 away from here and 2 away from there. Okay? So this is another measure of centrality. It doesn't have to do with shortest paths. It has to do with just how many hops do I have to make between every node and every other node. For example, if I want to spread information rapidly, I don't want to be at C, I want to be at B, because then the information will spread to all of these other nodes in a faster way. And then there are many, you know, scenarios that you can construct that sort of distinguish these metrics from each other. I mean, node B was the most central node for each of those metrics that I gave you. So it had the highest degree, it had the highest betweenness, and it had the highest closeness. But in this particular case, you know, node A, for example, has a much higher, or not a much higher, but it has a higher degree than node B. But node B is both more central and closer to the rest of the network. Okay? So you could basically say, hey, node A is very central according to its degree, but node B is very central according to its betweenness. Okay? So there's another way to define this, which is using eigenvectors. So eigenvector centrality basically extends the concept of a degree, where you basically make xi proportional to the average of the centralities of all of the neighbors of I. So you basically now have a measure that basically say, how central are my neighbors rather than how central am I? And then lambda can be some kind of constant that basically weighs how much you know, information going to flow in this particular fashion. And then the matrix vector notation of this would be, you know, x, basically, you know, this xi, instead of having a sum, you can just have a matrix multiplication between A. And then in order to make the centralities non-negative, we can select the eigenvector corresponding to the principal eigenvalue. And we're going to see a lot more with eigenvectors and eigenvalue. Basically, this is one way of computing, you know, all of these paths in a much more efficient way by sort of transferring that through the matrix transformation. And again, you can revisit the slide after we've talked about eigenvalues and eigenvectors, which is actually coming up now. So basically what we talked about in part 2 is basically all of these different measures of network centrality. So local centrality metrics such as degree, betweenness, and closeness, and then global centrality metrics, you know, which is very related to the Bayes rank algorithm that you use every day when you Google something. So let's now review a lot of tools that can be used in the study of networks when you have this matrix representation of networks, and basically some basic linear algebra that will be very helpful for any kind of application. So some of the most important aspects of linear algebra is the ability to compute eigenvalues and eigenvectors. So you can think of a matrix as a transformation of a vector. So this is a transform that you can apply to that vector. So a vector can be, you know, any kind of data point in a particular multidimensional space. And then a matrix is effectively an operation on that space. It's a transformation of that space. So you can apply that transformation on any kind of point in that space. And there are transformations that basically will rotate some points. There's transformations that will sort of scale and stretch and so on and so forth. And there are some particular vectors that are such that if you apply that particular transformation on that point, which is basically the vector endpoint. So, you know, a vector is basically starting at the origin and going to some coordinate in that multidimensional space. The transformation of that particular point or of that particular vector sometimes results in a linear scaling of that vector onto itself, into just a scaled version of itself. Okay? And in that particular case, we can basically talk about the eigenvalue, which is basically the case where the matrix transformation is just a linear scaling of that vector. Okay? So basically, eigenvectors are such that when I apply that particular transformation to this vector, it's akin to simply scaling that vector. And that scaling value is the eigenvalue of that vector. Okay, so here's one example. So I have, this particular matrix, which is a transformation on any xy. It will basically return some kind of, transformation of xy. Specifically, 6x minus 2y will be, the first point, and then 4x, plus 0y will be the second one. And then when I apply that matrix to many, many different vectors, it will transform the vectors to point in a different direction. But when I apply it to the vector 1, 2, it is the same as simply scaling 1, 2 by scalar. Okay? Who's 100% with me on this? Raise your hands. Awesome. Great. So basically, this is, a very, very fundamental property that basically allows you to now start learning all kinds of things about matrices and vectors. So basically, how many eigenvalues are there at most? So, this is equivalent to sort of solving this particular equation. So you have, this particular matrix, the operation, basically you're trying to find the V, the vector V, such that that operation becomes a linear scaling. And this is equivalent to basically in, matrix algebra, just taking that transformation S, subtracting the scaling transformation by the identity matrix, and then multiplying that with vector V, and then ending up with 0. And that has a non-zero solution if this particular element is actually 0. And then this is an mth order equation in which lambda can have at most m distinct solutions, which are effectively the roots of the characteristic polynomial. And this can be complex. These solutions can be complex, even though, the particular matrix S is real. Okay? So you can solve this to basically find what are the eigenvalues and the corresponding eigenvectors of a particular matrix. So here's one such example. So we have matrix S, 3 to 0, etc. It has eigenvalues 3 to 0 with the corresponding eigenvectors. And the reason for this is that this matrix is already decomposed into, it's effectively SVD form. So in every eigenvector, S acts as a multiple of the identity matrix, but as a different multiple for each eigenvector. So for any vector, say, 2, 4, 6, this can be viewed as simply a combination of the eigenvectors, 2v1, 4v2, 6v3, where you're basically multiplying these corresponding vectors accordingly. Okay. So therefore, matrix vector multiplication, such as Sx, can be written in terms of the eigenvalues and eigenvectors. So Sx, which can be basically transformed into S times each scalar operation on the corresponding eigenvector. And I can transform those into the corresponding eigenvalues because, when I have these eigenvectors, any kind of transformation S on each of these eigenvectors is effectively just a scalar, which corresponds to the corresponding eigenvalue. And basically, these can be written as 2SV1, 4SV2, 6SV3, and then SV1 is simply lambda 1v1, because when I transform the corresponding eigenvector, I just get a scalar with the corresponding eigenvalue. Who's 100% with me so far? This should be review for many of you. So who feels that they actually learned something? Yeah, good. Okay, or at least we refreshed something. Good. So even though X is an arbitrary vector, the action of S on X is determined by the eigenvalues or eigenvectors. So basically I can, transform any kind of arbitrary vector into the corresponding action on the eigenvalues by expressing that vector in terms of the corresponding eigenvectors. So the effect of small eigenvalues is actually small. So what's kind of nice here is that, if you scale this accordingly, you're basically learning what are the dimensions of this where the effect is the smallest. And then you can use that to start learning about, how to decompose matrices. So for symmetric matrices, eigenvectors for distinct eigenvalues are orthogonal of each other. So, if I have a particular eigenvector and then these eigenvalues are not the same, then the dot product of these vectors becomes 0. And then all eigenvalues of a real symmetric matrix are actually real. So for complex lambda, if S minus the eigenvector the eigenvalue times the identity matrix is 0, then S can be the transpose of S, and then that means that the lambda is in the real. It's not a imaginary number. And then all eigenvalues of a positive semi-definitive matrix are non-negative, and then the corresponding mathematical formulations here. So here are some examples of eigenvalues and eigenvectors. So basically, if I have this particular matrix, which is both real and symmetric, then if I take that matrix minus lambda I to identify the eigenvalues, then it is the lambdas that basically set this equation to be 0, and then the corresponding lambdas are just 1, because 2 minus 1 square is 1, and then 3, because 2 minus 3 is minus 1, square is 1, minus 1, 0. So basically 1 and 3 are the non-negative and real eigenvalues corresponding to these eigenvectors, and then the corresponding eigenvectors are actually orthogonal of each other. So 1, 1 points to the right of the space, and then 1 minus 1 points exactly the opposite. Okay? So this is just a practical example that gives intuition about the previous property. So that now takes us to, again, something that's extremely fundamental in computational biology. When you have these extremely high dimensional data sets, but they're sampling observations which are extremely high dimensional from effectively a lower dimensional system, then you can actually start decomposing those to actually find the true dimensionality of your system. And that's, basically finding the principal components of variation. And, PCA is effectively based on this eigen-decomposition and understanding what are the, true dimensionalities of your system. So basically, if your matrix S is a square matrix that has m linearly independent eigenvectors, so a non-defective matrix, so that when I find all of the linearly independent eigenvectors, they're actually distinct from each other. Otherwise, that matrix doesn't have the true dimensionality that you think it has. It's a defective matrix. Then there exists an eigen-decomposition such that S can be expressed as a set of columns, each of which is your eigenvector, a set of rows, each of which is the same eigenvector, and then a diagonal matrix of eigenvalues. So the columns of U are the eigenvectors of S. In some cases, these will be the rows as well when U and U transpose are the same as U and U minus 1 and the inverse of U. Most of the time it will not be the case, but in some cases that we're going to see, this will exactly be the case. And then the diagonal elements of lambda are basically the eigenvalues of S. So, lambda is effectively the diagonalization of a set of scalars, which has zeros everywhere except for the diagonal elements, which are the eigenvalues. So why is this diagonal decomposition possible? So if U has as eigenvectors these columns, then we can write SU, which is the transformation of each of these vectors by the matrix S. So this S transformation of every one of these vectors basically is the same as applying S to each of those, but applying this S to any one of those is the same as a scalar scaling of that corresponding vector. So I can write that as lambda v1 all the way to lambda vn. And then I can take those lambdas, and instead of having them to the left, I can put them to the right. So I can actually diagonalize that matrix from lambda 1 to lambda n, and then take the scalars out. It's exactly the same operation, whether I multiply them this way or the other way. Everybody with me on this step? And basically, that gives me U lambda. So SU is the same as U lambda. So if I multiply both sides by U inverse, then I get SU U inverse, which basically cancels each other. And then U lambda U inverse, which is exactly this decomposition that I was talking about earlier. Okay? Who thinks this is kind of cool? Raise your hands. Awesome. Great. Okay. So basically, this is how I can actually create this decomposition. So given these eigenvalues and eigenvectors, I can basically create such a decomposition for any matrix S. So where I have the set of V's here, the inverse of that there, not the transpose, the inverse. In some cases, it will also be the transpose. And then the lambdas and the diagonal. So here's one example. So here's the same matrix that we had before. It has eigenvalues 1 and 3, eigenvectors, you know, the diagonal to the bottom and the diagonal to the top. And then, you know, I can basically decompose this as U, which is the matrix of eigenvector columns, which is 1 minus 1, 1, 1, straight from this. And then if I invert that, this is, you know, the inverse of this, such that u minus 1 is 1. So basically, if I multiply this with that, it is the matrix such that if I apply this transformation and that transformation together to something, it becomes the identity matrix. So it's the inverse transformation of that in this dimensional space. And then, you know, I can basically write this decomposition of S as u lambda u minus 1 as basically the eigenvectors, the eigenvalues, and then the inverse of the eigenvectors. Everybody with me? Awesome. All right, any questions so far? So we've covered, hopefully, a lot of refreshers for those of you who've taken linear algebra before. But, you know, this is, you know, just a very, very helpful operation to then be able to do all kinds of stuff with it, as we're going to see in the rest of the lecture. So if we divide u and multiply, you know, u minus 1 by square root of 2, I basically end up with this. And that basically allows me to now scale these in a particular way that allows, you know, this particular matrix to be exactly the transpose of that particular matrix. So there is such a scalar, square root of 2 in this particular case, that allows me to transform this particular equation such that these rows happen to also be the eigenvectors themselves. Okay? And then that's basically when I multiply these by a particular scalar. So Q minus 1 is the same as Q transport in this particular case. Q transport Q is the identity matrix, and that's an orthogonal matrix where every column has unit length and is perpendicular to all the others. Okay? And basically what I had to scale these by is a number such that these scalars actually become, you know, unit length when you multiply it. Okay? So this is symmetric eigen-decomposition. So basically, if S is a particular symmetric matrix, and there exists a unique eigen-decomposition with the transpose in this particular case rather than the inverse, where Q is orthogonal, and then Q minus 1 is QT, and then the columns of Q are actually normalized eigenvectors. So what you have to do is simply scale them in a particular way that allows this particular product to end up being the identity matrix. So the columns are orthogonal, and then everything is written. So singular value decomposition is exactly this. So it's basically for any m by n matrix A of rank R, there exists a factorization known as singular value decomposition, and that's the basis of principal component analysis, or SVD, that basically transforms A into 3 matrices, an m by n matrix, an n by n matrix, and then an m by n matrix, where the columns of U are orthogonal eigenvectors of A transpose, in this particular case. The columns of V are orthogonal eigenvectors of A transpose A, and then the eigenvalues are the eigenvalues of A transpose A. So instead of you know, working directly on A, I'm now going to be doing an SVD on A transpose. And then the singular values are effectively these sigmas, which are nothing more than the square roots of the corresponding eigenvalues. So that's the difference between singular value decomposition and eigen decomposition. Instead of working on A, I'm working on A transpose. Okay. So if I have you know, a dimensionality that doesn't quite match, then, you know, if I don't have a square matrix, then I can still carry out this SVD decomposition into the U, S, V transpose matrices, understanding that some of these columns are simply going to be, you know, zeroed out in this diagonal matrix. So here's an example of singular value decomposition, where basically I have these 2 vectors, and then, you know, in this particular case, notice that the singular value matrix is ending here with zeros at the bottom. and you know, this is SVD, or non-diagonal matrix, basically MN matrix rather than MM. So what's really cool here is that we have now constructed what we need to carry out low rank approximations of complex objects. So SVD can actually be used to compute optimal low rank approximations. So if I have a 100-dimensional space and I want to approximate it into a 20-dimensional space, then I just carry out SVD exactly as we saw, and I just take the top 20 eigenvalues and I zero out all of the other eigenvalues. Okay. So, you know, the particular approximation problem that I'm solving is that I want to find AK of rank K such that I'm basically minimizing the distance between A and, you know, this low rank approximation under this Euclidean norm. And then Euclidean norm is simply this particular object. You're basically squaring all of the absolute values. So I'm basically minimizing over X with rank K this particular distance operation. So basically, I'm basically finding the matrix K of rank, sorry, the matrix X of rank K, which provides the best possible approximation to matrix A. Then this is simply this SVD approximation where I zero out all of the eigenvalues from K plus 1 to the rest. Okay. So then the solution via SVD is basically, you know, use U, VT as before, but now instead of having this S matrix of all of the singular values, I'm going to take the first K singular values and I'm going to zero out all of the remaining singular values. Okay. And that basically has the properties such that this actually minimizes that error, and that error is, you know, exactly this. Okay. So everybody with me on sort of this low rank approximation? Who's following so far? Raise your hands. Awesome. Good. So a big question is, great, I have this 100-dimensional object. How many dimensions should I project it into? I have, you know, I've measured gene expression patterns in 100 conditions. How many true conditions was I truly observing? So what I can do is basically say, well, let's look for, you know, this lower rank approximation, but how do I choose the rank to which I want to approximate it? Do I choose 20 or do I choose 40? One way to do that is to basically look for non-random eigenvalues and eigenvectors. So does the network, does the corresponding matrix look like a random matrix? And does it have some non-random, or does it have some non-random structure? And one way to do that is to basically exploit this Wigner semicircle law, which basically is the distribution of singular values of a random matrix. And what you find is that if this is a random matrix, it will follow this kind of circular distribution, also known as a Tracy-Widom distribution, the probability distribution of the largest eigenvalue. So this is the probability distribution of the largest eigenvalue of a random matrix. So what I can do is remove lower rank eigenvalues and corresponding eigenvectors, or singular values, I'm going to corresponding singular vectors, until the remainder of the matrix effectively looks like this. Okay? So this is the distribution of eigenvalues of a non-random matrix. And you can see here that this is the random component, which follows this circular distribution. And then these are the non-random components. What you want to do is define a p-value for every PC, and then consider the significant PCs in the analysis. So here's one example of structural inference using SVD. So basically I have M experiments, I have N genes, here's the corresponding matrix. I can calculate the singular values, which are basically, you know, all of the meaningful combinations of genes by experiment. And then this is their distributions. And then this is the number of eigen experiments, how many experiments did I truly do, and how many genes did I truly observe. And what you can see here is that based on the structure of that matrix, most of the non-randomness, if you wish, was in the first 3 genes for the first condition, and all the rest was, much lower rank. So, you can think of the Eigen experiments are basically having most of the information coming from the first experiment and relatively little from the rest, and then most of the information coming from the first 3 genes and very little from the rest. Here's another example where I basically have, all of this information coming from the second and the fifth experiment. But the second and the fifth experiments are in fact very similar to each other. I can actually, when I do the singular value decomposition, I can basically recognize that and then, zero them out accordingly. All right, so we've basically just reviewed a lot of linear algebra in relatively little time. About eigenvectors, eigenvalues, singular vectors, and how I can do an eigenvector decomposition and a singular vector decomposition based on this A versus A transpose. How I can generate lower rank approximations by basically taking only the K, largest eigenvalues and corresponding eigenvector, or singular values and corresponding singular vectors. And then just this Wigner semicircle law that basically tells you that that the rest of your matrix is effectively random if these distributions actually start obeying the the circular rule. Okay. So now let's basically principal component analysis has been basically about SVD. So basically SVD is how you carry out PCA. Let's now talk about sparse PCA. So basically, I carry out a bunch of gene expression analysis, gene expression measurement. Say I measure 20,000 genes in each of 100 conditions. And here the number of genes is much, much larger than the number of conditions. And what I want to do is carry out, principal component analysis on the expression data. So basically, every eigen gene is expressed only in the corresponding eigen array, the corresponding eigen expression level. So basically, I transform my matrix of conditions versus genes into effectively 3 matrices: a matrix of eigen genes, a matrix of eigen experiments, and a matrix of eigen combinations of which eigen gene is expressed in what eigen experiment. Okay? So what this basically allows me to do is basically say, what is the relationship between my experiments? So if I have an M by M matrix of experiments, this tells me just how similar are these experiments to each other. Truly understand, the unique information of those experiments. What are the eigen genes? Basically, what are the unique dimensionalities of variation of the corresponding genes? And then how are these eigen experiments, how are these eigen genes active in each eigen condition? So one drawback of PCA is that the principal components, the eigen experiments, are linear combinations of all of the variables, all of the genes. So every PC corresponds to a loading vector, and there are just a lot of loadings. So basically the loadings are the coefficients corresponding to the variables in the linear combination. So that basically means that even though I was able to sort of successfully decompose it, these eigen experiments are experiments over all 20,000 genes. And what I would like to do is learn experiments over a subset of those genes. So one way to do that is basically to carry out sparse PCA. So the idea is to reduce the number of explicitly used variables, the number of genes, by modifying PCA so that the principal components have relatively sparse loadings. So basically not every gene is involved in every one of them. So this basically allows us to write PCA as an optimization problem similar to a regression problem. And this basically uses Lasso, which stands for least absolute shrinkage and selection operator, which was developed by Tipshirani in '96. And that allows both variable selection and regularization, and it produces sparse model. So the result is you have a set of modified principal components with sparse loadings. So here's the linear regression problem. You basically have a set of input variables, X, a set of response variables, Y, which are some function of X with some random noise, and then a set of regression coefficients for every one of those. And then you have a multivariate linear model that basically gives you a function from all of the input variables to all of these transformations based on the regression coefficients. So you can express this as a linear regression problem where basically the output variable y is some kind of scaling of a bunch of training data and some kind of error. So there are n observations and p predictions, and the goal is to estimate the coefficients and basically specifically those betas. So the least square solution would be you have some kind of training set, you measure what is the residual sum of squares in 2 dimensions or in multiple dimensions, and then the objective is to find the set of parameters that minimize this residual sum of squares error given your data. The sparse PCA solution is to basically use exactly the same training set. Here's the residual sum of squares as before. And then the lasso penalty is a penalty on the coefficients, and specifically how many of those coefficients are non-zero. So what we'd like to do is basically find beta under lasso that minimizes both the residual sum of squares error and the lasso penalty on these coefficients. Basically, the pro is that the lasso continuously shrinks the coefficients towards 0, and it produces a sparse model using this variable selection method. The cons is that the number of selected variables is limited by n, by the number of observations. For example, the microarray expression data, the number of arrays is usually typically much, much less than the number of genes. So it selects only one of the highly correlated variables, and it doesn't care which one is in the final model. So what we'd like to do instead is allow it to select multiple of those. So the way to do that is to basically use different kinds of regularization. So you can use that, for example, linear regularization simply looks at the sum of absolute weights of all of your coefficients. L2 regularization or ridge regularization allows you to basically sum up the squares of those. So this is a linear function that's a quadratic function. And then the pros and cons of the corresponding functions is that for the linear function, the pros is that it's more robust. The outliers don't matter any more than the small differences. But the cons is that it has a less stable gradient ascent and multiple solutions. Whereas the L2 always has one solution, it is more stable, but the cons is that it's actually less robust. So the square differences are much more strongly affected by these outliers. So how do you combine the advantages and disadvantages of both L1 and L2 regularization? That's where the elastic net comes in. So basically, it combines the lasso penalty and the ridge penalty by basically, including both and effectively combining both the lasso and the ridge regression. So the limitation of the lasso is removed by the ridge constraint and vice versa, and all the variables are actually included in the model. And then you can select a group of highly correlated variables, once one variable among them is selected. Where lasso selects only one of these variables. It doesn't care which one. Here you're now selecting a group of these variables. So basically what sparse PCA does is that constructs a regression framework in which PCA can be reconstructed exactly. And then it uses either lasso or ridge or elastic net penalties to basically construct modified PCs with sparse loading. So here's how you construct PCA in a regression framework. Basically, the idea is that every PC is a linear combination of the P variables, and the loadings can be recovered by regression of the PCs on the P variables. So these are the P variables, this is the training data. Your response variable is basically this combination using this eigen-decomposition of the corresponding coefficients. And then you can actually do that to solve the corresponding framework. Okay. So here's some examples. If you have a bunch of data points, you have 10 variables, you use some model to generate the data where there's only 3 hidden factors. And then when you decompose that using sparse PCA, what you find is that it will group the variables into these 3 components. And there will be 4 variables associated with the first component, 4 variables associated with the second component, and 2 variables associated with the third component, as opposed to only picking one such vector for each of those. So, this is the corresponding loading where you basically now taken, non-zero loadings for PC2, non-zero loadings for PC3, in each case allowing you to find a combination of 4 different input variables. All right, so here we are. We basically looked at overall networks, network centrality, and then some basic introduction to linear algebra with eigenvectors and singular value decomposition, and then this sparse PCA or SPCA with different penalizations for these variables. And now we're going to switch gears again and basically start talking about network communities and modules. So the goal there is to basically find groups of genes or groups of variables that behave similarly. So the goal is to basically predict the functions of unannotated genes. For example, I'd like to, predict the function of a gene that I know nothing about when I see that it behaves similar to a bunch of genes whose function I know. So this is the concept of guilt by association. So I can have a co-expression network, I can have a protein interaction network, I have a co-regulatory network. And if I know the function of the orange genes here, I can say perhaps something about the function of the gray gene here, and similarly there. So, for example, I can have an iterative classification algorithm where I start with an initial assignment of labels, and then I iteratively repeat, updating the relational attributes and re-inferring the labels. So if I have some unknown genes, the labels of the unknown genes will be influencing each other, and then will need to be inferred jointly. So one way to do that is to exploit the modularity of biological networks. So basically, if you have a graph with densely connected subgraphs, I'd like to be able to automatically discover these densely connected subgraphs. So if I have genes within a module, I will assume that these genes within the module are involved in similar functions and co-regulated, and therefore, if many of the genes in that module are of particular function, I can make some inferences about the genes whose functions are not previously annotated. And this can be achieved using different types of graph partitioning algorithms. So, for example, there's Markov clustering algorithms, there's Gervan-Newman clustering algorithms, and an algorithm that we're going to look at today is spectral partitioning. So what that is going to do is utilize these eigen properties of the connectivity graph of these networks to actually partition the networks in optimal ways into pairs of subgraphs each time. So the way that you define these communities can be very subjective. So you can have a very densely knit community where every component is connected to every gene is connected to almost every other gene. Or you could basically say, hey, these sub graphs are in fact connected together into a larger subgraph, and so on and so forth. And that definition of a community can be subjective. So basically, there's no right answer as to what it should be. For example, should the yellow one be cut here, or should be, should it be cut at this level? At one extreme, every component, every connected component of the network is a community, whether or not it's connected by a single edge. In another definition, densely connected subgraphs are in fact what defines your community. Then you can define these community criteria using different ways. So the criteria can depend on the task. So community detection methods can be divided into 4 categories. For example, you can have node-centric community detection, where every node in a particular group satisfies certain properties. You can have group-centric communities, where you're considering the connections within the group versus the connections outside the group as a whole. And the group has to satisfy certain properties without zooming into the node level. You can have network-centric communities where you partition the whole network into several disjoint sets. And you can also have hierarchical community building, where you basically build a hierarchical structure of your communities, and then you choose a cutoff at some point. So, for example, you could go extreme and say, I want every node to be connected to every other node and in my community. So that's a clique, a maximally complete subgraph in which all of the nodes are adjacent to each other. So nodes 5, 6, 7, and 8 form a clique. But it's actually NP hard to find the maximum clique in a network, even though straightforward implementations can find cliques. They're very expensive in their time. So how do you find the maximum clique? In a clique of size k, every node maintains degree at least k minus 1. Yeah? Yeah? For example, yeah. So node-centric would basically be that all of these nodes have some kind of property in terms of their connectivity to other nodes or their distance from the rest of the graph and so on and so forth. Whereas edge-centric or group-centric would basically be, so basically the group-centric would be that, you know, the connections within the group, as opposed to node-centric, which can be, so if you only look at connectivity, I guess the node-centric can be quite similar to the group-centric. It basically tells you something about that node and how it relates to other nodes. If you have additional functional information, for example, the node properties could simply be that all of these nodes are highly expressed genes in this particular condition, or something like that. You could define them based on their properties. Yeah. So you could basically say these are all of the genes involved in creating a particular complex, and then these are the genes involved in creating the complex and the three complexes that they interact with. And here's all of the genes that are involved in sending other proteins to that complex or receiving signals. And Exactly. And then histone proteins associated with that, or polymerase 2, and the initiation of transcription, because all of those are actually coupled to the translational rate and stuff like that. So basically, when you have, it's just different layers of similarity in that organization. Exactly. Any other questions? So basically, what we can notice is that if there's a clique of size k, every single node must have degree at least k minus 1. So then any kind of node with degree less than k minus 1 cannot be possibly included in a maximum clique of length k. So we can recursively apply this pruning procedure, sample a subnetwork from the given network, find a clique in the subnetwork using some kind of greedy approach, and then suppose that the clique is size k. In order to find out a larger clique, all the nodes with degree less than k minus 1 must be removed. So that allows you to very rapidly prune nodes from your network to sort of facilitate your algorithm speed. And then you repeat that until the network is actually small enough to be able to construct this. So instead of finding a globally optimal network, you're basically first sampling, and then you're saying, well, yeah, great, I found something in that sample. Let's now use that as a way to prune other nodes that I don't need to look at. So the concept here is that many nodes will be pruned as, many biological networks and social networks follow power law distributions for their node degrees. So that basically means that, the probability of finding a node of very high degree is actually, very rapidly decreasing in a power law setting, an exponential decrease. And that basically means that if I sample randomly, most of the time I'm going to pick nodes with low degree, and that will allow me to sort of start constraining my network. So if you sample a subnetwork of, with nodes 1 through 9, for example, we find a clique of size 3. And in order to find a clique of size more than 3, we remove all nodes with degree less than 3, minus 1 equals 2. So basically, I remove nodes 2 and 9. And that basically means that I can then remove nodes 1 and 3. And I can remove node 4. And then I'm left with that, which basically gives me the clique in a rapid fashion. So here's a group-centric community detection. So basically density-based groups. So the group-centric criterion requires that the whole group satisfies a particular condition. So for example, that the density of the group is more than, say, some parameter alpha, given a threshold. So the subgraph, G induced by the subset of vertices V and the subset of edges E that are incident in these vertices Vs, is basically gamma dense quasi-clique if it satisfies this particular equation, whereby the number of edges times the number of vertices minus 1, squared, is, at least gamma. And thereby the denominator is the maximum number of degrees that I can possibly have. And then a similar strategy to that of cliques can be used, where you sample a subgraph, you find the maximum gamma minus dense quasi-clique, gamma dense quasi-clique of size Vs, and then you remove the node with degree less than the average degree, and then this is what that gives you. So network-centric criteria basically need to consider the networks within all the connections within the network globally, not just locally with their neighbors, but globally. So what we want to do is partition the nodes of the network into disjoint sets by either clustering based on vertex similarity, or using some latent space model, some kind of multidimensional scaling, or using some block model approximation, or using modularity maximization, or what we're going to see now, which is spectral clustering. And what spectral clustering allows you to do is basically take the adjacency matrix, as we saw before, of the network, then construct the Laplacian of that matrix. And what is the Laplacian? The Laplacian is basically the degree matrix minus the adjacency matrix. So the degree matrix is effectively what is the degree of every node, so K1 through Kn. And then you're subtracting from that the adjacency matrix, which is, all of the Aij's here. And what that gives you is, so if you have the degrees here and then the adjacency matrix there. Okay? Everybody clear with what the Laplacian is? So if I basically have an adjacency matrix, I can compute the Laplacian of that graph by this particular operation. And then I can start figuring out how to modularize that matrix. Here's my adjacency matrix. Here's the Laplacian matrix. All I've done is simply add the degree of every node on the diagonal. And the eigen-decomposition of that effectively gives me vector. So basically, the Laplacian can be eigen-decomposed in exactly the same way. You basically have the eigenvectors here, the eigenvalues here, and then the inverse of the eigenvectors. This is not SVD, this is not singular value decomposition, this is eigen-decomposition. So what's really cool when I do that is that the very first eigenvector of this Laplacian matrix is effectively pointing all in the same direction. The second eigenvector effectively splits the network into the maximally distinct sets of connectivity patterns. So these guys are all pointing in one direction, 1, 2, 3, 4. And then these guys, 5, 6, 7, 8, are all pointing in the other direction. So what I can do is take the Laplacian of a matrix, eigen-decompose that Laplacian, and then take the second eigenvector, which effectively splits that matrix in, that corresponding graph of that matrix into two. And that allows you to now identify, all of the subcomponents in specific directions. So basically, after this decomposition, then you have 2 pointing in one direction, 3 pointing in the other direction, and so on and so forth. Okay? So the first eigenvalue of the Laplacian matrix is always 0. The second eigenvector of the Laplacian matrix characterizes an optimal network partition. So the Laplacian matrix basically characterizes the number of edges between groups. If I have group 1 and group 2, the ith component of the Laplacian is basically, the diagonal term of the Laplacian, then the off-diagonal terms of the Laplacian, which is effectively the number of edges going out of each group. So remember how half of them were positive and half of them were negative. This is basically what allows you to partition that network in that particular way. So you basically choose a vector S to minimize the error term, and then a trivial solution is S always equal to 1 and the error is 0, but a non-trivial solution is S being parallel to the second eigenvector of L, and then, why? Because this network modularization by decomposition is effectively, projecting S over the eigenvectors of the Laplacian, and then, there's the second eigenvector of that effectively characterizes the partition, and then the vector S should be some integer valued projection. And then that's sort of what allows you to identify things by partitioning at the network level. Yeah. Correct. Based on the, basically the intuition here is that what you're trying to do is identify the main modes of directionality of your network. Basically, that's what singular decomposition does, singular decomposition, Eigen-decomposition. Basically, if you have this Laplacian, what it basically says is, if I were to pick two particular directions on which I sort of spread my network, these will be the two dimensions along which I go. So you can basically think of it as sort of the dimensions of variation of the connectivity patterns. So if I have a connected network, it basically, tells you, and if I pull it apart, what it will basically do is sort of define the two dimensionalities, the two opposite directions along the dimension of maximal decomposability, if you wish. Does that make sense? All right, so we basically, talked about how to build these communities and modules. The last thing I want to talk about is how information diffuses on these networks. So you basically can define the closeness of two nodes in the network by using, either a weighted shortest path, but I'd like something that's invariant to the position of the edges on the path. So I can basically define a diffusion kernel given the adjacency matrix of a network, given the diagonal matrix of degrees, given the Laplacian, which is nothing more than d minus a. I want to define a diffusion kernel matrix that effectively diffuses according to some diffusion parameter beta. And that is, raising this Laplacian minus beta into some exponent. So basically, K is that kernel that allows information to diffuse through the network. So this is a matrix exponential, not element-wise exponential. And then the intuition is what you want to do is write the Taylor series for matrices. So basically, exponentiation of this minus beta Laplacian matrix is effectively, spreading out information from every node to every other node in a decreasingly, in a sort of constantly decreasing fashion, which basically gives this geometric decay, exponential decay. So if I have this particular adjacency matrix and this particular degree matrix, and the Laplacian matrix is again, d minus a, and then there's an interpretation of what we're doing via a random walk. So basically, I can randomly walk according to the transition probability from every node to every other node, whereby the transition probability from any node to any other node is constant, some constant beta. And the higher the beta, the faster I diffuse through the network. The lower the beta, the slower I diffuse through the network. And then the remaining probability is simply the self loop that have, flow probability from every node, and then 1 minus 3 that times that flow probability, because all of the exit probabilities have to sum to 1. So Kij is effectively equal to the probability of the walk that started at I, being at position j after an infinite number of time steps. Okay, so that's the closed form solution of this infinite decay function. So, a lazy random walk would be a Markov model where the transition probabilities are beta and 1 minus the degree times beta. And then the transition matrix is this. And after one step, we're there. After n steps, we're there. And what I would like to do is reduce the time step 1 to delta t and have some kind of diffusion parameter beta delta t, and then taking the limit as delta t becomes 0, this effectively gives me that kernel matrix of expansion. So one interpretation of that is a stochastic process where I have a random variable, walking through these particular nodes. And there's a time evolution operator, which is basically this, some kind of covariance. And then I'm reducing the time steps to infinitesimally small. And that basically gives me this particular covariance. So one way to think about this is that every single time I have a set of correlations that I've measured between pairs of genes, there's a direct correlation that I measure always. And then there's a set of indirect correlations that basically come from just the fact that if A is correlated with B and B is correlated with C, then I'm going to find some kind of correlation between A and C. So if I have a true network, as shown here, because 1 leads information to 2 and 2 leads information to 3, then I'm going to end up in my observed network having some information flowing from 1 to 3 directly. But in fact, what I would like to do is reverse this information, this process of diffusion. I'd like to sort of figure out what are the direct edges from that. And one way to think about that is that the observed edges are nothing more than the direct edges plus a set of indirect edges that are obtained by repeatedly multiplying that matrix by itself, which is effectively, the transitive closure of hops of step 1, hops of step 2, hops of 3 steps, hops of 4 steps, all the way to infinity. And that is the same, effectively, diffusion process. So when this converges, this basically ends up with, some kind of this, infinitesimal series, which can only converge if this maximum eigenvalue of this network is less than 1. So we need to scale the network somehow in order to achieve that condition, basically achieve this convergence of the network. And then this beta is the decay rate. With a small decay rate or a large decay rate, these initial priors persist longer, shorter. So given this transitive closure of the network, you can basically inverse this function and effectively deconvolve these indirect effects by simply writing the closed form solution of this observed network as G dir times 1 minus G dir inverse, which is effectively the closed form solution of this. And that basically means that I can inverse that equation, basically observe to infer the direct edges from the observed edges. And when we applied that, basically we found that indeed this was allowing us to improve the quality of many of these networks. So anyway, what I wanted to give you today is a set of tools that you can use to study all of these biological networks of many different types. We talked about many different types of networks. We talked about measures of centrality. We talked about how we can decompose these matrices corresponding to these networks, how we can carry out PCA and sparse PCA, how we can discover communities, and how we can actually understand diffusion of these networks and these kernels. And on Thursday, we're going to talk about deep learning. Thank you.